Okay, I think we have everything set up. Let's double check. Thank you for your patience. I know it's not the most convenient time for you in the UK, but
No, it's not too bad. It's only 8 p.m. so
we're
could be
thinking
worse.
about maybe moving the meetings a little earlier, but finding a time when people can come is always. I mean, the problem is if we did something that was really convenient for Europe, then the people on the west coast of the US are in trouble.
Yeah.
Zoom can abolish the distance, but it can't abolish time zones.
Yep.
How is the weather? Have you had the heat in Scotland that you've had, that everyone else has had?
Yeah, it's been quite nice. I mean, nice for Scotland, which is probably still cold for other places.
I mean, the first time I went to Dundee was, what was the year that there was the huge heat wave where thousands of people died?
Oh, really?
Trying to remember the year, maybe 2000, 1998, something like that. And Dundee was very comfortable. It was 70 degrees, sunny, beautiful. We went to London, it was 115 degrees. And on the tube, people were collapsing because it was so hot. Nobody had air conditioning, of course. So
yeah, we don't need it really.
No, up in St. Andrews on the ocean. You were very.
Yeah, well, I actually live in Dundee, so.
So you still live in Dundee, okay?
Yeah,
so,
yeah.
But it's the same weather, really. Not so far.
Yeah.
No, you're not very far inland in Dundee. You're still on the water.
Yeah, that's true.
Of course, the flip side is it never gets really cold. It gets wet and raw, right? When it snows, and I remember snow in Dundee is pretty miserable because it's so wet and windy, and but you don't get the kind of cold, because you can grow things in your gardens that we can't grow here because it gets much colder here. And
yeah,
of course,
it's usually just
dry.
mild and rainy.
I mean, I remember always visiting Case Weir's house. The garden just astounded me what he could grow in his garden. Everything was green all the time. It was beautiful.
Mm-hmm.
So is this paper published yet? I had the link I found was still to archive.
No, the paper I'm going to talk about today has been published. But the PhysiCell model that it's part of has not been published yet. It's still just preprints.
Yeah, rapid is a relative term. But no, because could you put maybe put the link in the chat, because when I searched you, I wanted to have the link to the published paper, but the Google Scholar only pulled up the bioRxiv paper, didn't pull up the published version. So I want to share that with the audience, make sure that they have the updated one.
Yeah, that's the link. Should be
great. Okay. I'll reshare it because Zoom does something funny, which is people who join can't see the chat before they join. So we'll give people a few more minutes. Hi, Tom. You look very intense.
I'm just reading through the pile of emails.
Can't hear you.
Test. Test. How about now? How about now? No?
Boy, we maybe really should be moving the time, because we have more people in Europe than we do in the US today. Is the rail strike affecting you in Scotland as well as in England?
Yeah, I think it is. I mean, not me personally, but I think a lot of the trains have just not been running the last few days. So it's pretty bad for those that do depend on the train.
I mean, it's up at D and St. Andrews, you'd be in a car anyway, and so the traffic wouldn't shift. I think if you're around London, where most people probably use the trains, and if everybody gets knocked into a car, the traffic is probably pretty horrible.
Yeah, I can imagine.
Lorenzo had the good sense to go on holiday with his family in Italy to avoid the strike.
That's why I'm not showing up too much today. Hello. Hi, Fiona. Nice to meet you.
You too.
We'll give it another minute or two. But my experience is that during the summer, people will watch these things offline more, because it's everyone is on holiday somewhere, except me. I'm always at the same desk. Never.
The benefits of everything being online is you can go and watch it later and not have to worry about it too much.
I mean, our talk last week was on experiments on pyroptosis.
Oh, was it?
It's a good segue.
Okay, so maybe I don't need to explain too much about what it is. That's quite
well, it's always better. You don't know it'll be the same people in the audience.
Yeah. That's good timing though.
Yes, also UK speaker. It was Claire Bryant from Cambridge.
Okay, I think I know the name, yeah.
Her interest is comparative immunology. It lines up that the chickens and cats don't do pyroptosis. So you get different inflammatory patterns to infection. Well, I think we'll probably then get started and see if people join in. Some people will watch offline, I'm sure. Welcome everybody to the June 23rd meeting of the working group. We have one speaker. We've been hoping for a long time that she'd come and speak to us, so we're very glad that she's here today to join us and let us know about her work on modeling SARS-CoV-2 induced pyroptosis. As always, I need to remind you that this meeting is live streamed and recorded and archived for future viewing, so please be aware of that. I do mildly edit the recordings to trim the chit chat and those sort of things. The recordings are there. So I remind you, as always, Tom and I are currently co-leads of the working group. We thank you for your participation and suggestions. Jim Sluka remains the person to go to for getting your papers and ideas up on the website. If you have recent papers or announcements, job openings, and so on, please do post them to the wiki, and we will do our best to make sure that those are publicized appropriately. Lorenzo has been doing a great job booking speakers, and Joel Vanin from my group has volunteered to help Lorenzo out starting this week. So if you see emails from Joel, you'll know that he is one of the team trying to bring this series together for everyone. We thank Joel a lot for being willing to join us. As always, we have our communication channels. They only work if you use them. In particular, we have that MSM viral Twitter feed. We'd love it if people would retweet. We have our IMAG MSM wiki page. It has a lot of nice information on it. Please post your papers, ideas, suggestions to that. We have a LinkedIn page. You're welcome to post to that as well. There is a Facebook listing as well. And of course, our YouTube archive of recordings, which we would love to have you help publicize. Again, we have that Twitter feed, MSM Viral, easy to remember. Please do help us get the word out and help us use it more effectively. Are there any specific announcements for this week that people would like to make? I would say that we have a steering committee meeting tomorrow at 11 a.m. If you think you should be invited to the steering committee and you haven't gotten an invitation, please let us know so we can send it to you. Gary and Chase want to announce their recent publication in Frontiers in Systems Biology, drug development digital twins for drug discovery, testing, and repurposing. This is the second paper of Gary's in the area of digital twins in the past few months, coming out of working group discussions. I guess Gary is in surgery today, I think he said, so he's not able to join us live, but I hope he'll tell us about that work soon. I also would remind you that we have this nice Google form that makes it easy for people to suggest speakers. And I will paste that link into the chat a little bit later so that we have that. Are there any other announcements? Tom, Jim, Lorenzo, anyone else? No? Okay. Next week, it's almost July 4th, so we're going to take a break on the 30th of June in honor of the 4th of July holiday. Maybe we should be honoring European holidays more, but we'll do the US one this week. July 7th, we have industrial speaker, we hope, who's doing very interesting work in standards and artificial intelligence. And then Tom Knudsen from the EPA who is a very famous computational toxicologist, is going to speak on the 14th of July. As always, we would love to have speaker suggestions. If you've spoken before, that doesn't mean you can't speak again. As you've heard me say many times, feel free to suggest your friends, the papers that you've read that you liked. Could be experimentalists, could be journalists, could be context, could be standards, could be modeling methodologies, could be experimental techniques, could be corporate, development, medical. Be flexible, think about who you'd like to hear, and we'll do our best to get them on the line for you. And we really do appreciate your help. Your students, your postdocs are welcome to speak. This is a very open forum. So please do help us come up with some good names. Lorenzo's done a great job of trying to line people up. And one thing more is that if you do suggest names and you know the people, usually it's a little bit more effective if you make some contact yourself in terms of getting people to respond. So please help us keep the meeting going and keep it lined up. Okay, I don't want to take any more of Fiona's time. We're very pleased to have Fiona MacFarlane from St. Andrews University, who's going to be telling us about her single-cell mathematical model of SARS-CoV-2 induced pyroptosis, which, as I was mentioning, is a great follow-on to the talk that Claire Bryant gave last week on species differences in pyroptosis. And I will turn the screen share over to you.
Thank you. And just share my screen. Okay. Can you see my screen? Yeah. Okay, perfect. So thank you very much for the invitation. My name is Fiona MacFarlane. I'm a postdoc at St. Andrews University within the UK, working within the mathematical biology research group. And the project I'm going to talk about today was part of the PhysiCell coalition, which I'm sure some of you are aware of, which aim to look at modelling COVID-19 infection in lung tissues. But the talk I'm going to focus on is a simplified model that was implemented into that fuller model. So we'll start with the simple model and then look at how we integrated that into the multi-cell model as well. So I'm going to give a brief introduction to pyroptosis, although as I've just heard that you got a nice introduction to pyroptosis in your last talk. We'll then look at the single cell model, which is an ODE model of pyroptosis, and then look at how we integrated that model into the wider multicellular model. So pyroptosis is a fast and highly inflammatory form of cell death, and there's several things that this form of cell death is characterized by. So one important step in the pathway of pyroptosis is the formation of an inflammasome within the cell, which is made up of NLRP3 proteins. And what this inflammasome does is it allows for the cleavage of pro-caspase 1 into an active form of caspase 1. This caspase 1 then allows for the cleavage of inflammatory cytokines like interleukin 18 and interleukin 1 beta, and also allows for the activation of gasdermin, and then this gasdermin can form pores on the cell surface. And these pores then allow water and other things to enter the cell and cause the cell to swell, and also allow for the release of these cytokines and other things from within the cell. And eventually the cell will swell so much that it eventually will burst. So hopefully this video will work, but I found this nice video of some in vitro experiments of cells infected with a virus that are undergoing pyroptosis. So these are immune cells in an in vitro setting. So if we focus on this cell here, we see that it's getting slightly bigger, and then eventually we see this kind of dramatic bursting of the cell. And we see the neighboring cells start to burst around as well. And one important feature of pyroptosis is that there can actually be a bystander effect as well, where once one cell goes through pyroptosis, the inflammation and the cytokines released by that cell can actually initiate pyroptosis in neighboring cells as well. So that's another important feature that we're going to focus on today. So why do we want to study this form of cell death? So as I said, it initiates inflammatory responses. So for example, high levels of interleukin-1 beta that are released through this process can promote this bystander effect and cause other cells to initiate this form of cell death. The cells will also release interleukin-18, which can promote the recruitment of immune cells as well. So in a normal, healthy working immune system, there would be negative feedback that would regulate these immune cell responses and cytokine levels, so that this would be a controlled immune response that is trying to protect the tissue from whatever is causing this pyroptosis, so the virus, for example. However, in cases where we have a weakened immune system or there's something else going on there, then there can be dysregulation in this and the immune system can overcompensate. And this can lead to things like cytokinestorm, where we have this unregulated levels of cytokines within the cell or the cellular environment. So the main reason that we want to focus on this form of cell death is it has been shown that SARS-CoV-2 virions can initiate the inflammasome formation and cause pyroptosis in both epithelial and immune cells. So therefore it's relevant in the case of COVID-19, where it's been shown that this form of cell death is something that can occur in severe cases of the disease. It has also been suggested that targeting this form of cell death and some of these pathways would be something beneficial in helping to treat COVID-19 cases. So one important thing to note is that if pyroptosis can be delayed or inhibited, then the cells will switch to a non-inflammatory form of cell death, for example, apoptosis. So through treatment, if we can prevent pyroptosis from happening, then we can reduce the inflammation in the cellular environment. So I just want to give a brief overview of some of the biology underlying this before we look at how we model some of these interactions. So as I said, one key step of pyroptosis is the formation of an inflammasome. And this inflammasome is made up of NLRP3 proteins. And these proteins start out in an inactive state and then can become activated through some signal, where that signal can be calcium signaling or potassium signaling, or also reactive oxygen species. And once these NLRP3 proteins are in this active state, they can then bind together or oligomerize together to form this NLRP3 wheel. And this wheel is going to be the base of the inflammasome. So once this wheel has formed, then the other two molecules involved in the inflammasome can start to bind to this structure. So our other two molecules that we're interested in are ASC and caspase-1, which will be pro-caspase-1, and then once it's activated will be caspase-1. So once we have this NLRP3 wheel, the ASC can bind to the NLRP3, and it forms this structure where we have lots of ASC on the inflammasome. Then once this ASC is bound, then the pro-caspase can bind to the inflammasome and can be cleaved off and become caspase-1. So this inflammasome structure is what initiates a lot of the pathways in the pyroptosis pathway. So what we want to do is we want to look at developing a model to capture these key mechanisms involved in pyroptosis in a single cell to start with. So we're going to be looking at an ODE model, and then after I've looked at the ODE model, we'll look at how that then relates to a multi-cell model. So we're looking at a single cell. So on the top, I'm going to have the kind of pathways that we're interested in, a kind of biological level, and then on the bottom, I'll put the equations. I might not go into too much detail on some of the choices for the equations, but please do ask if there's anything that's not clear. So each cell will have receptors on it that can then recognize different signals. And one of the signals that it can recognize is things like viral load of SARS-CoV-2. So once we get some signal, what happens is nuclear factor kappa B that's in the cytoplasm of the cell then moves into the nucleus of the cell. And we want to track the levels of NF-kappa B within the nucleus, and we do this through this function, where we're considering the change in time to be what it was at the start, and then some exponential function. And the reason we take it of this form is we want to be able to control the shape of that NF-kappa B peak. So as I said, this signal is going to be on if we have, for example, a high viral load or interleukin-1 beta through this bystander effect. However, for the ODE model, this only relates to the process starting or not starting. So once the NF-kappa B is in the nucleus, it can promote the transcription of NLRP3 in its inactive form, and can also promote the transcription of pro-interleukin-1 beta. So we take into account both of these terms. So for the rest of the components, we're going to have ODEs for these. So for our inactive NLRP3, we take a term where this F1 is dependent on the levels of NF-kappa B within the nucleus. And we take this to be a Hill function form to represent some binding. We take a similar form for pro-interleukin-1 beta as well, with just different rates of this transcription happening. So as well as these two components that are produced within the system, within the cell, there will be adequate levels of free ASC molecules, pro-caspase-1, gasdermin in its inactive state, and pro-interleukin-18. So now what we want to do is then look at how all of these relate and how we can then start to look at describing things like that inflammasome formation that I was talking about at the start. So with our NLRP3, we're interested in three states of this protein: the inactive state, the active state, and then once it's bound into that NLRP3 wheel. So we take that into account by using this term NLRP3O, or oligomerized NLRP3. So this is the NLRP3 that is bound together in that structure. And for each of these components, we use an ODE. So for inactive, we have this production term that we've already talked about from the NF-kappa B transcription. We have decay of NLRP3 in its inactive state and in its active state as well. We then have this kind of activation process dependent on this second signal, where the inactive NLRP3 can become active. And then we have this binding process where the NLRP3s bind together to become oligomerized NLRP3. So once we have this NLRP3 in the oligomerized state, that's when we have that base of the inflammasome formed. And once we have that base of the inflammasome formed, we can have binding of ASC in its free form to become ASC in its bound form. So we take just a simple equation where we just consider the binding of that ASC to the NLRP3O, and we take a function F2 of this form. So the reason that we take this function is that we want there to be some threshold level of NLRP3 required before the ASC can bind to that inflammasome, as we need that structure to be fully formed before the binding can occur. So once we have this ASC that's bound, then pro-caspase-1 can start to bind to that inflammasome structure as well. And again, for the pro-caspase-1 and the active caspase-1, we just take these terms representing that binding between caspase-1 and bound ASC. So I should have said all of these Ks are just rates of these things happening. So these will be parameters within the model. So once we have caspase-1 in its active form, it can then cleave gasdermin into its N-terminal form, so gasdermin N. So we just, for the gasdermin, we have this one term, which represents that cleavage process, where for this function F3, we take this to be a Hill function of caspase-1. And once we have this gasdermin in its N-terminal form, it can start to form pores on the cell surface. And these pores then allow our interleukins to leave the cell, and will also allow water and other things to enter the cell. So for our interleukins, we're interested in three states of these. We're interested in the pro state, the interleukin in the cytoplasm, and then the levels outside the cell once it leaves the cell. So we're going to take ODEs for each of these components. So for our pro-interleukin-1 beta, we again have this transcription term dependent on NF-kappa B. We have the cleavage process from pro-interleukin-1 beta to interleukin-1 beta in the cytoplasm that's dependent on caspase-1. So this F3 function is the same caspase-1 Hill function from the previous slides. And then we have some decay term of pro-interleukin-1 beta. In the cytoplasm, we'll then have the interleukin-1 beta that's been cleaved, some decay, and then also we'll have the interleukin-1 beta is leaving the cell dependent on the amount of gas dermin N that we have within the cell. For our external interleukin-1 beta, for the moment, all we take is this term, which represents the interleukin-1 beta coming out of the cytoplasm into the external environment. We're not necessarily interested in what happens to that once it leaves the cell for this ODE model. But when we integrate this into the wider model, we'll be interested in what happens to those molecules once they leave the cell. For our interleukin-18, we use very similar terms, except we don't have this production term or decay term. So we just have the cleavage process facilitated by caspase-1, and then the process of the interleukin-18 coming out of the cell through these pores. So as I said, with these pores, what can also happen is water and other things can enter the cell. So what we're also interested in is how the cell volume will change over time. So we take an ODE for cell volume, where this is just a function of the level of gas dermin in the cell. So we have this ODE model. So this is all of the ODEs and the functions that are related to it. Don't worry about the detail here. But what I want to highlight with this is that we have quite a lot of parameters within the model that then we need to find values for. So we parameterize the model using published experimental data where possible. And when we couldn't find values for certain parameters, what we did was we fitted those parameters using a timeline from published experimental results. So from a few different experimental papers, what we did was we looked at the timings of different things happening or when we would expect certain things to happen. So for example, if we have a start time of 0 minutes, where we expect the virus to be recognized, for example, the signal will turn on. Around 10 minutes after that signal has been recognized, we would expect the nuclear factor kappa B concentration to peak in the nucleus of the cell. Around 43 minutes before the cell dies or ruptures, then we expect there to be enough NLRP3 within the inflammasome. So we expect that base to have formed. Around 13 minutes before the cell ruptures, the pores start to form. So that allows for the interleukins to start to leave the cell, as well as the cell to start to swell. And then around 120 minutes after the initial signal, we expect the cell to rupture. So this is a very fast process where it's in the matter of a few hours that the cell will go from initiating the process to rupturing. So I want to show some simulation results of this ODE model. So for example, here we have the concentration of different components of the model and the cell volume in the bottom right-hand panel just over time. So for example, our NF-kappa B, we have this peak of NF-kappa B around the 10-minute mark, and then we have this decrease back to the normal level. For NLRP3, we have the 3 states. It's inactive state, it's active state, and it's oligomerized state, where this last state is when it's bound in the inflammasome. So we can see the processes where it goes through these states, and then we see this kind of tailing off once the NLRP3 is within that inflammasome. Once enough of this NLRP3 is formed, then we see the increase of bound ASC, and then subsequently the increase of caspase-1 in the cell. So once this caspase-1 increases, then we get an increase in gas dermin, N, which then allows for the increase of external interleukin 1 beta and interleukin 18 as it comes out of the cell. And once the gas dermal pores form, we see this very quick increase in the cell volume. So from experimental data, it was shown that once the cell reaches roughly 1.5 times its initial cell volume, that's when we would expect the cell to rupture. So that's why we have everything ending at that time point. So obviously this model was fitted to the data, so we're kind of getting what we expect from the ODE results. But one thing we can do is we can then look at what happens if we change different things within the system. And one interesting application would be to look at things like inclusions of an inflammatory drug, an anti-inflammatory drug. So there's several anti-inflammatory drugs that target this process. So for example, tranilast or orodonin can both bind to activated NLRP3. So that prevents the NLRP3 from binding to itself to form that inflammasome structure, or it can delay the formation of that inflammasome structure. So we can include that into the model in a very kind of implicit way by just adding in some terms for a drug and NLRP3 complex. So we're going to consider that the drug and NLRP3 bind together at the rate K plus D, and then the complex can dissociate at the rate K minus D. We'd also be interested in the drug level over time, and then we would update our NLRP3 in its active state. The ODE for that term, that component, would be updated to add in this drug interaction. So we can look at what that does to the model by simulating different dosages of the drug, where we can then look at the different components or the outputs of the model. So for example, we're looking here at the drug and NLRP3 complex, where we expect there to be more of this complex if we have high dosage of the drug. Our oligomerized NLRP3, so that's the NLRP3 in the inflammasome, and we expect this inflammasome formation to take longer if we have more drug. And in fact, when we have this green line, so this highest dosage of the drug, then the inflammasome is not quite meeting the threshold level to allow for the binding of ASC. So that's why all the other terms, we just had this straight line for the highest dosage, where we've been able to prevent inflammasome formation. So we can see that having this drug, we can delay the effects of pyroptosis, so we can delay the formation of the pores on the surface by this gas dermal, or we can eventually get to a case where over this time scale, we can prevent that happening at all. So with this model, we have this ODE model that can capture pyroptosis or parts of the pathway of pyroptosis in a single cell. And we've parameterized this to match experimental data or to fit to experimental data where possible. And we've looked a little bit at the effects of this anti-inflammatory drug. So what I've not shown is in the paper, we do go into more detail about sensitivity analysis and looking at how sensitive the model is to each of the parameters, to try and get an understanding of which are the key parameters and which ones would perhaps need more accurate values to be sure that they're the correct ones. So what I want to briefly talk about is how we then take this single cell model and combine it with the multicellular PhysiCell model. So for those of you that aren't aware, PhysiCell is an open source physics-based cell simulator. And in 2020, in response to the COVID-19 pandemic, the PhysiCell group set up this SARS-CoV-2 coalition. So the coalition is a multidisciplinary team made up of biologists, mathematicians, and others from across the world. I think on the last paper, there were around 35 authors, but I think there's a lot more than that in the full coalition. And the aim of this group was to develop a comprehensive multi-scale simulation framework for SARS-CoV-2 infections in lung tissues. So there's several subgroups within this, and each subgroup focuses on a specific component. So the model at the moment is a model of lung epithelium that contains the effects of the virus, so things like viral spread and replication and release between and within cells. It also now includes immune response, so things like immune cells and how they would infiltrate and attack the virus-infected cells, as well as the effects of inflammatory responses. Tissue damage is included in the model, cell death, which is what we're kind of focusing on. There has been some work done on the effects of pharmacology and different treatments, and the effects of other areas of the body. So, for example, the inclusion of the effects of the lymphatic system have been included in the model. So the whole PhysiCell model is currently on version 6 of development, and the first 5 versions preprints are available along with all of the code in GitHub. So throughout the whole process, it's been a very open source and community-based idea, and the code can be played with and downloaded by anyone, and people can work on it together. So what I want to talk about today is how we implemented our pyroptosis model into this multicellular model. So we're going to scale back a little bit and consider the version 4 of the PhysiCell model. So this is a version before the immune system was added in. So in this system, we have epithelial cells, we have viral particles and the effects of the virus between cells, and we've got 2 forms of virus-induced cell death. If the viral load in a cell reaches a threshold level, then the cell can either undergo apoptosis, where the cells will reduce in size until they're removed from the system, or the cells can undergo pyroptosis. And the way that we allow the cell to go through pyroptosis is that it will go through the system according to the ODE model that I described. So when the cell undergoes pyroptosis, it will release interleukin-1 beta, and the interleukin-1 beta levels in the extracellular environment can also cause a potential bystander effect, where cells, if they detect enough interleukin-1 beta, can then undergo pyroptosis as well. So as I said, this version 4 of the PhysiCell model doesn't include the immune system or immune cells at the moment, but that's something we could then incorporate into this in future. So for each cell, there's going to be several rules that the cell will go through. So if the viral RNA load reaches a threshold value, the cell will enter pyroptosis with a probability of capital Pi, or it will go through apoptosis with 1 minus that probability. Or we're also going to include this bystander effect, that if the interleukin-1 beta levels reach a threshold value around the cell, then the cell will also enter that pyroptosis pathway. So once pyroptosis is initiated, it's modeled through the ODE model that I discussed. Then the cells will secrete interleukin-1 beta. Once secreted, this interleukin-1 beta will go into a diffusible field outside the cell, which can diffuse and decay within the tissue, and that can then cause a bystander effect in nearby uninfected cells. So as with the ODE model, once the cell volume reaches 1.5 times its initial value, then in the simulation, what we do is we just get the cell to undergo very fast apoptosis, and all processes cease in the cell. So I want to just show a couple of examples of this wider multi-cell model results and how we can look at pyroptosis specifically within that. So our initial condition is going to be this epithelial layer, where these are all healthy, viable cells in blue. We're then going to compare the viable cell levels with things like apoptosing cells. So those are cells that are going through apoptosis because of a high viral load. Cells that are undergoing pyroptosis from a high viral load as well in orange. And then also the cells that are undergoing pyroptosis from a bystander effect. So I just want to give an example of some results. So this is some simulations where we're looking at comparing this bystander effect. So in the first two columns, we've got the cell distributions at 30 hours and 36 hours. And in this final column, we've got the number of cells in each of these subpopulations over time. In the first row, what we have is we have that the threshold level of interleukin-1 beta required to initiate pyroptosis is a half of the kind of baseline value. In case B, it is the baseline value, and in case C, it's 1.5, the baseline value. So we're increasing the threshold required to initiate the bystander effect in each of these cases. So what we can see from this first case is in around 30 hours, we have a very large proportion of the cells that are red, which means that they're undergoing this pyroptosis via the bystander effect. And actually we end up with, at 36 hours, there's no cells left in the simulation. For the middle case, we have kind of equal levels of cells undergoing pyroptosis and apoptosis, pyroptosis via the viral load and pyroptosis via the bystander effect. In this case, there's no apoptosis. And in the final case, more cells undergo pyroptosis via the viral effects rather than the bystander effect. And that can be seen in the plots of the cells over time, where in the first case, we have a larger proportion of cells going through this bystander effect. And then going down, we see that this bystander effect takes a lot longer to take place in these later cases. So something else we can look at with this full PhysiCell model is comparing the effects of apoptosis versus pyroptosis. So what we have here is in case A, we have that all cells will undergo apoptosis and pyroptosis is not possible. In the middle case, there's a 50-50 chance of going through pyroptosis or apoptosis. And in the last case, cells can only go through pyroptosis as a form of cell death. So from the simulations, it's very clear that apoptosis is a lot slower of a process than pyroptosis, and we have a lot more cells left around the 36-hour mark in this case. We can also see that when we just have pyroptosis, we have this increase in the bystander effect, as well as obviously cells going through that pyroptosis, which means that the cells are removed a lot faster in that case. So with these results, we just wanted to highlight that we can look at specific aspects of this pyroptosis within the full PhysiCell model. Obviously, at the moment when there's no immune system in there, we're not getting the full dynamics. So what would be the next step would be then to look at the PhysiCell model that has the immune system included, and then look at how we can incorporate the effects of pyroptosis in that case as well. So we have this ODE model that captures pyroptosis in a single cell, which we've parameterized to fit experimental data and looked at things like the effects of an anti-inflammatory drug and performed sensitivity analysis to look at key mechanisms in the model. We then incorporated this into the multicellular PhysiCell framework to investigate the bystander effect a bit more and look at apoptosis versus pyroptosis. So what we could include also in the PhysiCell case is the pro-inflammatory effects with immune response. So as we said at the start, interleukin-18 that would be released by the cell as well through pyroptosis can promote immune cell recruitment. So that's one mechanism that we could link with the immune cell response in the PhysiCell model. So all of the work that I've talked about today was done completely joint with Sarah Hammis, who was at St Andrews University as well, but now is in Tampere. And the PhysiCell work was obviously done with the PhysiCell SARS-CoV-2 coalition as well. If you're interested in the pyroptosis specific things, Sarah and I wrote a paper that was published in Ames Mathematics that covers the ODE model and how we, a little bit about how we integrate that into the PhysiCell model. And the code for the ODE model and the PhysiCell stuff for pyroptosis are all available on GitHub. For more details on the full PhysiCell model, the version 5 preprint is available on bioRxiv, and all of the model details and code are available on the PhysiCell website. But I'd just like to thank you again for the invitation to speak, and I'm happy to answer any questions.
Lovely. Thank you very much. I'm sure there'll be questions. The floor is open.
Hi, Fiona. Nice to meet you. Thank you for your call or for your talk. It was very good. I particularly know a little bit more about your paper because I based on one of my projects that I worked with Glacier in one of his classes. So I have a intimate relationship with your paper, let's say, for the last semester. And it's very good. It's good. I can tell you, I could replicate just by your notes. So props to you. And but one of the things that are most important now that we're trying to work more and more with the difference of cell death and how the immune system respond, I think one of the important questions that I have is, do you have any data from in vitro studies that corroborate with your findings in the sense of epithelial cells producing as many IL-1 beta as expected from your model, and how much that affected the neighboring cells, or the need for actually immune cells to produce that meaningful amount of IL-1 beta?
Yeah, that's a great question. So at the moment, we've not really done much in the way of taking the model forward. And yeah, we don't have any direct links with data in that way. The only thing I would say is we did find some kind of evidence of some of the levels that would be expected to be released from the cell. So I think it was around 90% of the IL-1 beta that's produced is then released by the cell. But yeah, it would definitely be interesting to try and find something more specific, or try and work with experimentalists who'd be able to tell us the answer to that.
So I mean, the IL-1 beta here, you're talking about the not the pro IL-1 beta, but the cleaved IL-1 beta.
Mm-hmm. Yeah.
So I guess one question I had is your validation. I know it's in your paper, but just to go over it, the response of macrophages to infection, the response of the epithelial cells to infection is rather different. Did you think about the differences between the pyroptosis response in those two cases? And if so, what would your insights be?
No, we didn't consider that. We just considered pyroptosis in the infected epithelial cells, although it would be possible for pyroptosis to also be initiated in the immune cells. As I said, we've not really related it to the full immune cell model. So in the PhysiCell version 5 model, they've included immune cells, including macrophages and T cells. So it would definitely be interesting to then look at that kind of interplay between the immune cells and the pyroptosis pathways.
But it looks like potentially the level of IL-18 and IL-1 beta may be released, may be different depending on the particular cell type that's going through pyroptosis. So that was, and certainly your model could include that if you had experimental data to validate it. So that's great. Lorenzo.
Hi, thank you for the talk. It was really nice. Thank you. And mine is on the, you say, you show as a result that pyroptosis and apoptosis have different timing. And I wonder if this is emergent out of just the model itself, or if it's something intrinsic that you know already from data of assumption that one is faster than the other for some reason.
So it's something that's already been kind of shown by experimental data. I can't remember the exact timings of apoptosis, but we found, experimental data that suggested that pyroptosis takes around 2 hours, whereas apoptosis has been shown to take longer than that. And with the apoptosis, the PhysiCell model has apoptosis as an inbuilt system. So the timing of apoptosis is kind of related to experimental data, I think, in the PhysiCell model as well. So it's not emergent from the modeling side, it's something that was kind of already inbuilt.
Yeah, okay. So is this is because I'm ignorant. I don't know pyroptosis. I've ever heard about pyroptosis. It is a form of necrosis or something like that. So the cell becomes bigger and bigger and just goes out, explodes, without having the chance to do all the nice thing apoptosis helps doing.
Yeah. Well.
Thank you.
Well, so let me so maybe that wasn't clear. No, pyroptosis is a form of programmed cell death. It's not like classic necrosis where you lose ATP and the cell bursts because it doesn't pump its water. So it is controlled.
Yeah.
But it's more pro-inflammatory than apoptosis.
Okay.
Okay. Lorenzo, Joel, did you have another question?
Yeah, I have one more. It's about the gas dermin choice because clearly it controls how fast the cell will die. So your choice for how many was already inside the cell was purely based on the time frame that you needed for 2 hours for it to die, or do you find any evidence that none of it is produced? Cells have already established how many gas dermin is inside of it during pyroptosis?
Yeah, so I've not actually looked at the paper for quite a while, so I'm trying to remember. From what I remember, all of the components that we didn't include production of, we found evidence that there was an adequate level of gas dermin, for example, to be in the cell. However, in our model, we obviously we kind of normalize things, so we just aren't necessarily looking at actual concentrations of gas dermin, for example. It's just a sort of percentage level of how much we would expect. So I think it's set at 1 to start with, if I recall. So yeah, that's something that would obviously need to be improved upon, and to validate the model would be to actually look at specific concentrations of these components.
Thank you.
I maybe would like to follow up a little bit on that. Actually, connects to both Lorenzo's question and Joel's, which is, and I certainly understand why you did it the way you did it, but the oligomerization and then the complex formation, in principle, is a multi-step process. And you're collapsing that into a single response function.
Yeah.
And of course, writing ODEs is a pain when you don't know whether you have all the combinatorics of the possible oligomers. But do you think that the process of assembly is something that could potentially be significant to the way the system plays out as a whole? Or do you think that assembly is so fast that dealing with it as a single Hill function or a single more complex transfer function is sufficient?
Yeah, so with the oligomerization process, we did do, so for that ODE, it comes out as just an NLRP3 squared term. But what we actually do in one of the appendices of the paper is look at building that up. So if we have, say, M NLRP3s within the inflammasome and bind that up sequentially, then it does reduce down to this square term. But we're not interested in really, we assume that it's a very fast process, that we're not interested necessarily in each of those single steps. But we did think a little bit about that kind of binding sequentially rather than just a single term.
Because it's fairly intimidating to think about all the combinatorics of those inflammasomes. Right? You form your disc, then the inflammasomes conjugate with those other proteins, and then they aggregate into clusters. And so if you really wanted to do that, some rule-based framework like BioNetGen might be the way to do it, so that you don't have to worry about enumerating all of the species in advance.
Yeah, I think with this model as well, because we were building the ODE model to go into the PhysiCell model, so we didn't want to make it too complicated so that it would work nicely within the PhysiCell structure without being too difficult to integrate.
And I guess one thing else to come back to on the bio side is the mode of action of the bystander effect. Because you could be having a bystander death from DAMPs released by the pyroptosis.
Mm-hmm.
Or you could be having a bystander effect induced by the IL-1 beta or something else. And in your model, you're assuming it's the IL-1 that's doing the bystander killing.
Yeah.
You have, in the experimental literature that you were looking at, was there anything that indicated whether that was primarily the case? It's sort of hard to separate them. I guess you could dump IL-1 beta on a culture without any damps and see what happened. Somebody else in the room maybe would also have ideas about that, but I was wondering what your thinking was.
Yeah, that's a good point. For this model, we kind of initially talked to an immunologist who was working with the PhysiCell team, and she was the one who suggested that IL-1 beta would be kind of promoting this bystander effect the most. And I think there was a few references to that in the paper. But yeah, it would definitely be interesting to investigate that a bit more and see if there were other things that could potentially initiate that process as well, which I imagine there would be, because it's a very pro-inflammatory system, that there's probably more things going on than just these simple mechanisms.
And presumably at that point, you need more TLRs than just the one that you started with.
Yeah, in the model, we don't actually consider the structure of the receptor. That was just for descriptive purposes.
Again, I'm not trying to be critical. I think that the level of simplification is fine, but you always want to sort of ask the question, which of these simplifications would you unpack first?
Mm-hmm.
Other questions for our speaker?
I guess I have one last one. I don't know how they implemented the production of the virus on their package, right? But I assume that is infected cells produce virus and non-infected cells don't produce any virus, right? And it keep replicating. Have you noticed in any of your, because I believe you run this multiple times on your machine before you find the correct answers, right? Or what your model was showing, but did you find in any one of those that there was a control of the viral reproduction just by the bystander effect of pyroptosis happening on other cells? Meaning there was no count of virus after a certain threshold?
Yeah, that's something that I don't think we actually double checked. And that would definitely be something interesting to check. I'm not sure if that's something that the wider PhysiCell team would be doing as well. So with the PhysiCell model, everybody inputs into the big model, and then there's a specific team who then works with the full model to find different results. So we were only looking at a tiny aspect related to the pyroptosis stuff. But that would definitely be something interesting to look at. Yeah, thanks.
Yeah, I'm just commenting because it seems that that response, right, that type of death might serve some specific behavior, right, to protect somehow the tissue.
Yeah, I mean, that's the kind of goal process of pyroptosis, is it's actually a protective thing. because it tells the cells around that there's a virus there and tries to prevent the virus from spreading widely, but can then also lead to this over-activation of the immune response as well. So one thing also that we could also include is other kind of negative feedback in controlling the pyroptosis a bit more, because as you saw in the PhysiCell model, all the cells just die out. So there obviously needs to be some other processes in there to prevent that from happening.
And do you know what potentially are the anti-inflammatory signals that relate specifically to pyroptosis? Is it IL-10? Is it sort of standard IL-10, or is it something else?
Yeah, I think there's also interferons that are involved, if I recall correctly, which I think some of the interferons that are involved have been included in the wider PhysiCell model for other aspects. So that's again something we could tie in, that they could then prevent some of this pyroptosis or control this pyroptosis as well.
Other questions? I was going to ask one follow-up question just on the experimental data sets. You talked about the timings at which events occur as constraining. Are there any detailed time series of any of these components, or is that something that's fundamentally impossible to measure in cells, live cells?
Yeah, I'm trying to remember the data that we used, because it was quite a while that we did this. I think from what I remember, there was some time series data, but we had to take data from several papers to construct a timeline. So it wasn't all just one piece of data, which obviously means it's not as reliable if it's coming from different sources. But yeah, that would definitely be something to look into, as well as maybe trying to find something a bit more concrete on that and time series data on that.
Okay, any other questions for our speaker? No, well then I'll thank you very much for a great talk. Appreciate you being willing to speak to us, and look forward to seeing how these models develop as they get integrated and expanded in the future. Thanks so much.
Thank you.
You.
Good.